Good morning. Let's start. I'm going to recap a little bit about haploid selection. There's a couple more things I wanted to show you, and then we're going to add mutations in. And so yesterday, I was talking entirely about models where things breed true. By that, I meant that an individual of a type, whether that be a COVID lineage or a big A allele, had offspring that were the same type. And so today I want to start the process of talking about when things don't breed true. And I want to briefly talk about the diploid model of selection, which doesn't breed true, as well as add mutations. And the mutations are going to be the main subject matter of today. But before I do, I just wanted to recap that yesterday I talked about two kinds of models. Ones that treat time as continuous, so time is just passing, and ones that treat time in discrete units like years or days or whatever your units are. And I pointed out that you get very similar forms of change over time. Both of these models have the same kind of relationship to the variables. And if you plot them, they would be very, very similar to one another as long as the selection and the fitnesses are similar. And let's just recap what the assumptions are. So this is saying over time, we can predict what will happen. And it doesn't, these models, as for these breeding true haploid cases, it doesn't depend on the number of types. So I'm mainly focused on only having two types, big A and little a, but the same process holds for any number of types. And I showed that in a little proof when we talked about the ratios. It doesn't depend on the ecology. But I mentioned that that has a big if. And that big if is that ecological interactions affect everybody equally. And the math makes it clear in what way I mean everybody equally, right? If we're talking about fitness in a discrete time model, everybody has to have a reduction in fitness by the same factor. And so that it makes it clear what has to hold. Density dependence has to affect everybody equally. Otherwise, you have to model both processes. But I started yesterday's lecture saying that evolution by natural selection was a special force. It was a special one of the evolutionary processes because it helped explain the match between organisms and their environment. And I did all of this P and Q wizardry, but I never really got back to, well, is the population in any way getting closer to the optimum in the current environment? So I'm going to do one little proof, so you're not responsible in any way for the proof, but I think it helps see where these things come from, that the mean fitness rises over a generation in this model. And then I'm going to expand the model to deal with diploids, just briefly. Okay, so here is the claim I want to make. I want to show you that the mean fitness from one generation to the next, these deltas almost always refer to changes. So the mean fitness in one generation minus the previous generation. I want to prove to you that that is always positive, or it might be zero if it's not changing, but it's never negative. And I'm going to use this discrete time model to do that. First, I have to say, what's the mean fitness? The mean fitness is the fitness of everybody in the population weighted by their frequency. This part, that's the mean, this part in the denominator is the mean fitness. And we call that W bar. And that's because it's the fitness of everybody, the big A types and the little a types, but we count up how many big A's we have, measured by P, and how many little a's we have, measured by Q. So this is the average over that population. Okay, so that means I can, in the next generation, I'm going to say that the mean fitness of the next generation is equal to the fitness is weighted by their frequency in the next generation. And so that's the mean fitness in the next generation. And then we have the mean fitness in the current generation, which just has T's in place of plus ones. Okay, any questions? There's a bunch of ways to do this, and the nice thing about math is it kind of doesn't matter how you do it. You'll get to, if you don't make mistakes along the way, you're going to get to the right answer. I'm just going to do a fast way for this proof, but it's not the most intuitive. The fast way for this proof is I'm going to gather terms that involve the same fitnesses, W big A, and I have P T plus 1 minus P of T. And I'm going to gather all the things that involve little a, and I get QT plus 1 plus minus Q of T. So this term represents the change in P. This term represents the change in Q. And so here's my little trick that makes this proof a little faster. P and Q always sum to 1. So if P goes up by a certain amount, what will happen to Q? By how much? By the same amount, right? So delta Q has to be equal minus delta P, because there's only two types that I'm modeling here, and that I can use that relationship. So then that means that we have a delta P in all of these terms, and in front of that we have a Q minus W big A minus W little a in front. So that, that can be positive or negative. So, I mean, this part can be positive or negative. That part can be positive or negative. So I'm not done with my proof. I need this delta P. And so what is that delta P? Well, I'm going to replace that delta P with my PT plus, I'm going to go back now and replace delta P with PT plus 1, which is W big A E of T over, I'm just going to call it mean fitness for now, but so that's PT plus 1. And then I have minus P of T. So this whole parentheses is the delta P written back out. I need to put this over a common denominator. So this P of T, I'm going to multiply and divide it by mean fitness. So I do that all the time. Multiply by 1, but the tops and the bottoms have the things that I want. This is putting things over a common denominator. So that leaves me with one step that I need to do, and that is clean up that numerator. So cleaning up that numerator, I have, I want to keep the board clean, so I'm going to use it right here. I have W big A P of T, and then I have minus P times the mean fitness. W big A P of T plus W little a Q of T. All right, and then I do the same trick I did before. I pull together everything that involves the same fitness, because if anything factors, they'll factor. So I have W big A, P of T, and then I have a 1 here, but I have a minus P of T there. Those are all of the things that involve that fitness. And then I have all of the things that involve the other fitness are minus W little a, P of T, Q of T. I stare at this equation for a little bit. It's easier to factor if I recognize that's just Q of T. So what does this all factor to? Eraser. I'm trying to replace this part now. I'm hearing it PQ times we have to do the W's. What was that? W big A minus W little a in here. Right. So the change in allele frequency from one generation to the next, and all of that was divided by the mean fitness. The change is proportional to the differences in fitness. That makes sense. If there's no differences in fitness, there's not going to be a change in allele frequency. If P is 0, there's no change in frequency. If Q is 0, there's no change. But now take a look. If I expand, these fitnesses are there and there. I'm going to take this one and put it inside. And while it was not obvious before whether mean fitness could go up or down, take a look at this. It's the difference in fitnesses squared. It cannot be negative. P is a frequency. It cannot be negative. Q is a frequency. It cannot be negative. It could be 0. When would it be 0? When the fitnesses are equal, or if there is no, only one type, no genetic variation. Otherwise, we have now proven that the mean fitness change from one generation to the next is positive. But to some extent, this is kind of the easy case. There's only one thing going on, and that is selection, because everything is breeding true. The next case I want to just briefly touch on is the diploid model. The diploid model is a little bit different because we have two types, and I want to include sex and segregation. So we have the introduction of a second process, this selection, but then we have meiosis and the coming together of gametes, the segregation and mating, and that's a second process. Why is that so? Why am I even emphasizing that? Well, let's just work through what I'm going to model. I'm going to have 3 genotypes. We're going to do it in discrete time. And so we have fitnesses of those 3 types. And I'm going to start a model with at the gamete stage. And I'm going to count. This is where I'm going to census, and I'm going to count that there's P of the big A gametes, sperm and eggs, and Q of the little a types. Then I'm going to walk it through and make those gametes come together. Let's see, there's going to be, I just want to make sure I do this in this. I think, it's okay. They're going to come together to make diploids. There's no selection in this phase, so those gametes come together to make diploids, but I'm going to add selection here in the diploid phase. Okay. So if the gametes come together, I could have a big A sperm or little a sperm. And the chance of that is P and Q. And then I also could have a big A sperm or little a egg, sorry. Sperm, it's not 2 sperm, it's a sperm and an egg. Those are my choices. I like these are called decision trees, and basically from every node you figure out the probability of every possible type of event that could happen. And if you nest them like this, like combining a sperm and an egg, then the probability of a combined outcome, like a sperm that's big A and an egg that's big A, is just you multiply the probabilities of every route to that outcome. And so we have a P squared chance of getting a big A and a big A together. We have a Q squared chance of getting a little a and a little a. And there's 2 routes to making a heterozygote, this route and that route. It doesn't matter if the sperm or the egg carries the big A allele. So summing those 2 up, we have 2 PT and Q of T. So this is the famous Hardy-Weinberg proportions. I don't like to overemphasize it because it really is dependent on this assumption of randomness. By that, I mean, these P's are the same as these P's. There's no differences in selection between males and females. There's random mating, and so which sperm you choose determines which egg you choose. And the world isn't always like that. There can be biases. For example, maybe big A sperm are more likely to combine with big A sperm. And these decision trees allow you to model those more complicated cases. But for now, we're going to keep it simple and just have random mating. So that's at the beginning of that we've just made our babies, our diploid babies, and they're about to be subject to selection. So these frequencies will change. In proportion to their fitnesses, in proportion to their survival. And let's just focus on survival, but the same model would work if it's fertility. These are frequencies. If you sum these three frequencies up, it's P plus Q squared, that's 1. These don't sum up to frequencies anymore. Once I've weighted them by their fitnesses, we've got less of some types. And so we have to divide by their total sum to get frequencies back out. We have to divide by the sum. And that sum is, again, the mean fitness is the fitness of each type times the frequency that we find that type in the population. All right. Now, I just want to raise an issue. And let's go, extreme examples are really helpful when you model because they help you see what's going on and whether your equations are right or wrong. What if the homozygotes have no fitness and the heterozygotes have 100% fitness? What would happen after after a round of selection? So I'm just saying that these ones are 0. This is an extreme case. What population composition would I have after selection? 100% heterozygotes. Yeah. If they bred true, I'd have kids that were 100% heterozygotes. But they're sexual. So sex selection has just built up this really strong genetic association between big A and little a, and now they're about to go through sex and break it apart, bringing the population back into Hardy-Weinberg in the next generation. And so what's the outcome of these combination of processes? Selection building up types that are more fit, and segregation kind of breaking them apart and recreating a population in Hardy-Weinberg. All right. So there's just a couple more steps that we need for this. I've gotten our adults now after selection, that's this line here, but I haven't made gametes from them yet. So to make gametes from them, what I need to know is what fraction of each of these gametes will be big A bearing sperm and eggs. So what fraction of big A homozygotes will be a big A gamete? 100%, 1. So I just take them all and I add them. All of their gametes are big A. How about the heterozygotes? Half. And how about these homozygotes? 0. Yeah, and so all I need is half of the 2, PQ, the 2 disappears, and I just have P, T, Q of T, W, heterozygote. Okay. So this, and then the mean fitness is given there. And that maps us from one generation to the next. It's equivalent, it's similar to this, but you can see now we have 2 types in the top and a mean fitness. How many people want to guess? You know how here I solved this saying I took the ratio P to Q and I said we could go back one generation, then the next, and then the next, and get this general solution over any amount of time I want. This could be 1,000 generations later, and I just plug T in. Do you think this model, it kind of looks similar, has a general solution? How many people think yes, it looks similar, should have a general solution? What? Yeah. Maybe some special cases for the fitnesses, but let's keep it general that for any fitnesses. And now, how many people think that there's no general solution? How many people? One. How many people have no idea? Okay, fair. The trick that I used yesterday, divide P by Q, it doesn't cancel out the P's and the Q's. They don't all turn into ratios. And it turns out there's no general solution for this very simple model. We just gone one step further and took our haploid model selection, made a diploid. And still, we know how it changes from one generation to the next, but I can't just plug T into a formula and tell you where it goes. So you can approximate it, but there's no general solution. But there is one trick I want to, oh yeah, question. There are patterns, and yeah, we can look at some of those patterns. There's definitely patterns. And like yesterday, we said if W big A was greater than W little a, then the allele frequency just rose and rose and rose in the haploid model. And that's true here too, if we have W big A greater than W big A little a and greater than W little a. And those kind of things can be proven. All right, one last thing that I want to do with this diploid model, and then I'll show you some simulations of it. So even though I can't solve it in general, there is a way that simplifies this equation, and I want to just do this for a second. And that is if I factor out a P from the numerator, then in what's remaining, I have P of T times W big A plus Q of T times W big A little a. And then over the mean fitness. This thing here kind of looks like the mean fitness again, but it's the mean fitness of a big A allele paired with an average other allele in the population. If you know you have a big A allele, then the chance that it's paired with another big A is P, and then its fitness is big A's fitness. If it's paired with a little a, it's going to have the little a's fitness. So this thing here has a special name. It's called a marginal fitness. And I'm just going to write it as W big A with a dot. That dot represents, it's got another allele in it, but that other allele is chosen at random from the population. This W big A this marginal fitness is going to change over time because the P's and the Q's that compose it are going to change over time. But at any one generation, you can measure this average or marginal fitness. And if you did the same on the denominator, what you would find is that you would have P of T times the marginal fitness of the big A allele plus Q of T times the marginal fitness of the little a allele. There's no need to do this. It's just kind of rewriting and grouping terms together, except I can use this now to prove that the mean fitness goes up or is zero and never declines just by my former proof. Because now every place you see W big A and W little a in here, it's the same formula as I had for the haploid model, except it's got a little dot. And those fitnesses represent these marginal fitnesses. So the same proof for the haploid model holds now true too, that in a single generation, when we measure marginal fitnesses in that generation, the allele frequencies will change in such a way that the mean fitness goes up on average. It's not as high as it could go up, like our toy example. If the fitnesses are all zero for the homozygotes, the best that could happen is if the heterozygotes breed true and you only have heterozygotes. So the mean fitness doesn't go up that much as the two homozygotes are created by segregation. The sum total of the process of natural selection and segregation still leads mean fitness to go up from generation to generation. In the next generation, I'm going to have slightly different marginal fitnesses, but in that next generation, I'm going to still have that proof hold. Okay. I could have the screen down while they're doing that. And I think he said just say, wave your hand and say screen down, and it would come. But before any questions before this gets obscured, and only one screen. Yeah. Fitness. Yes. Does it also apply to making it non-random? Well, if the mating is not random, then you have two questions. Is the mating non-random in a neutral way, where it doesn't change the allele frequencies, or is there sexual selection preferring one mating type or another? And if there is sexual selection, that can counter natural selection, so the mean fitness due to natural selection can for sure go down. But you're asking a more general question, and I'm not even sure I know the answer to your general question. For any arbitrary neutral mating pattern, will mean fitness always rise? For selfing, it does, but I don't know necessarily. So, good question. That's where math would, you need to do the math. Okay. Yes. I can't quickly prove it to you, but what I can tell you is if you took P of t as a function, P of t plus 1 is a function of P of t, and then replace P of t as a function of pt minus 1, and replace it again, you have to do a little bit of simplification, but the algebra all works out. Here, if you do that substitution, and I now plug in P of t as a function of pt minus 1, it blows up, no factoring. And then when I plug it in as a pt minus 2, it's even larger. So that might be an exercise to prove to yourself that there is no simplification as you scale up. And you might also want to prove to yourself that if you divided P of t plus 1 by the equivalent Q of t plus 1, then the left-hand side is a fraction. It depends on the ratio, but the right-hand side does not depend just on the ratio. All right. Okay. Screen. I can do it. Left, right. Maybe I can do it. Come on, come on. Come on. It's not responding to me. Come on. Thank you. Just the right one. Just the right one.
This one, right?
This one I want down.
Yeah. Just I'll share the screen now. Go on.
Thank you. Thank you. Okay, there you come. Let's see. So Deepa shared some simulations with you, and I think I've got some that I posted on Slack as well. Wake up. Good. And these allow you to model any amount of selection that you want, haploid selection, with mutation, without mutation, diploid selection, and so on. And one of the things that this also lets you do is see not just what's happening to the allele frequency over time, but you can also see other things, like how the mean fitness changes over time. Oh, this is mean fitness by the allele frequency, mean fitness over time. Oh, you can't see that yet. But it goes up, and you can play with these as much as you want, and you're not going to see in the haploid fitness that trajectory will look different, but it'll never go down. The mean fitness always goes up or stays the same. The allele frequency can go up or down depending on your fitness, but the mean fitness doesn't, as we proved it shouldn't. But then there was another question, which was with diploid selection. What happens? What are the patterns that we see? Wake up. And I mentioned that if there's the big A allele always confers higher fitness. The more big A's you have, the higher fitness you have. Then you have directional selection, and big A rises in frequency over time, always. If the big A reduces fitness, then it decreases over time. It's hard to see. I'm going to start up. If I start near 0 or 1, these dynamics always look, let me make it not quite, S-shaped. So these are, this is called a logistic function. It's S-shaped, and it's either S-shaped going down or S-shaped going up if selection is directional. Let's start this now down. And then if we looked at the mean fitness over time in this diploid model, does that. Okay, but there's 2 more orderings of importance. I said directional selection favoring big A, directional selection favoring little a. Deepa, I believe, categorized selection with 2 other types. The heterozygote has the highest fitness, heterozygote advantage or overdominance, or it has the lowest fitness underdominance. So if we have, let's go back to the allele frequency over time. Heterozygotes are more fit. Then it goes to this intermediate frequency. And if I started at a higher frequency, it goes down to that same intermediate frequency. So it goes to an equilibrium, and if we have time, or if you want to nab me, I'll prove to where it goes in this diploid model. So that's known. So there's an equilibrium at this point here. What's happening to the mean fitness over time in those 2 cases? Regardless of whether the allele frequencies are going up or down, it's the mean fitness is still going up. Or barely changing because I'm too close to the equilibrium. There we go. Okay, and how about underdominance? That's the last one I want to explore quickly. So now I don't want it too extreme. There we go. Now I've made the heterozygote the least fitness. And I've started at a particular place and the allele frequency went down. The only slider I'm changing is the initial allele frequency. And let's just move it up a bit. And 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 up a bit. You get that out of your way. And up a bit. And up a bit. Okay, what had just happened? If I started at a low frequency, it goes down. If I started at a high frequency, and a high enough frequency, it goes up. And right in the middle is another equilibrium. It's actually satisfies the same equation. And that equilibrium is a repelling point in this case. I mean, fitness goes up or, sorry, allele frequency goes up or down, depending on where it starts relative to that repelling point. What's the highest fitness in this example? The little a, little a. What happens to the little a genotype in this simulation? Goes to 0, it disappears. So it's not, again, evolution's not optimal. It doesn't see, oh, I need to go and jump to that other genotype because that's the optimal genotype. Instead, the big A allele rises in frequency and fixes. Mean fitness is rising that whole time. If I just go to mean fitness over time, mean fitness is rising, but it's not ending at the best height. Does anybody want to hazard a guess as to why the big A allele is rising, even though the big A homozygote's not the most fit?
Because the marginal fitness of A is higher.
Yeah, the marginal fitness of big A is higher here than the marginal fitness of little a. That's exactly right. And if you wanted to prove when they would exactly balance, it would be when the marginal fitnesses are equal. And that's how you solve for the equilibrium, or one quick way of solving for the equilibrium. Because if the marginal fitnesses are different in any generation, then one type's more fit and it'll rise. Very good. But in terms of the composition of the population, why is an evolution raising the big A, little a homozygotes? At least not much. Maybe that's the wrong way of phrasing it. Which is the least common genotype in this population? The little a, because I've started at such a high frequency, there's hardly any little a's, there's really hardly any little a's. Selection barely sees them. Yes, they rise up, selection favors the little a's within the generation, but that's such a little rise in frequency, it doesn't counterbalance the fact that the big A's are much more frequent, and the big A's are much more fit than the heterozygotes. So the fact that the big A's are really frequent and they're more fit than the heterozygotes means that overall we see a rise in the big A allele. This maybe is better seen here. I've got the three genotypes: big A, little a, and little a. And although all three are present, this is so rare that selection doesn't really see many of them. And the main fitness differences in this population are between the homozygotes and the heterozygotes. And it's underdominance, so the homozygote is more fit, big A, and it rises. And that drives the whole system up. Play with these dynamics if you want. I'll just write for your convenience what that equilibrium point is, if you're interested. If I have a place. I think this is it. I should double check before writing, but I think that's it. That's that equilibrium in this model. Okay, any other questions? So one more thing I wanted to re-emphasize is I've added one process, so aggregation, and yet we see that the mean fitness rises, and these populations, this diploid selection, should lead a population on average to be higher in fitness over time, a better match to the environment. But that doesn't mean evolution is optimal. It doesn't get you to the best genotype immediately, and sometimes it doesn't even see much of the best genotype. So evolution is myopic. It only really has a subset of the genotypes that are really determining the evolutionary changes that are happening. Ready to add mutation? Or any questions left on that? Okay. So next I want to have things not breeding true, and I'm going to add mutation. But before, let's just do a thought experiment where the big A allele confers a higher fitness. We'll keep it haploid at the beginning, and I add mutations to little a. What's going to happen to mean fitness over time if I start with a population all big A's? Now it's mutating a little bit. Mean fitness has to go down because I started it at the peak. Big A fixed. Everybody's got the highest fitness, and then I'm mutating it away. So immediately in adding mutation, I don't have to do any math. I know there are counterexamples. I know mean fitness can decline over time, and it just depends on where I start relative to this kind of pressure from mutations that Kavita talked about. And you can explore that. You can add mutations to this. Takes a second to load. I have to make the mutation rate really high to really see that mean fitness drop, but I can see it. And then I'm going to start with an allele frequency up at 1. So the big A is more fit than the little a, and yet the big A drops over time because of mutation. If I did a realistic mutation rate, it would still drop. You wouldn't see it on this graph though. So the reason this is still dropping below 1, not by the width of those dots. So if we increase the mutation rate, we can really see this push. And so now if I plot mean fitness over time, then we're seeing this claim that for the first time mean fitness can decline over time. Not a surprise. Okay. Mutations. As previous lecturers have emphasized, mutation is the ultimate source of the genetic variation needed for evolutionary change. So without a mutation, everything would grind to a halt. And indeed, in the models, the haploid models and the diploid models that we talked about, most of the time, big A fixes or little a fixes. So selection actually depletes variation. And as Mike pointed out, drift depletes variation. And so mutation, although a lot of it is bad for our health and bad for our fitness, without it, evolution, we wouldn't be here. Evolution wouldn't happen. So I think Deepa mentioned the different types of mutations. I'm just going to kind of talk about them in a general way, and they include point mutations, the stuff of SNPs. It includes structural mutations, inversions, chromosome fusions, fissions, duplications, and a whole genome mutations as well, polyploidization events of the whole genome. So there's many different types of mutations that can occur. And I think that this is review, but of course, it's not all mutations are equal. Some mutations don't hit anywhere near a gene or a transcription factor or whatever, are more likely to be neutral. Other ones do hit a gene, and even if they hit a gene, they may be silent or synonymous, not changing the amino acid, or they may change the amino acid. Hopefully that's review. And then if they're nonsense mutations, if they cause a stop codon and a premature stop to the protein. Okay, oh, before I do this, let's take a vote. How many people think that, okay, think about mutation rates across, I'm gonna show you a graph on the next slide, mutation rates across a lot of species that have been measured. And this is per base pair mutation rate. How many people think that that's gonna be highest in eukaryotes? And how many people think it's highest in prokaryotes? So let's go for the eukaryotes first. Highest mutation rate in the eukaryotes per base pair. Highest mutation rate in the prokaryotes per base pair. Anybody want to, vast majority of you, anybody want to offer an argument as to why the prokaryotes with these teeny little cells would have a higher mutation rate?
Assuming that they have less mutation.
Yeah. 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 So they're small cells. How much can they do? How much can they repair? Prokaryotes, lower mutation rate. It's exactly the opposite. Eukaryotes have a much higher mutation rate. So this is showing the DNA error rate, the base substitution rate per nucleotide site per generation. And the reds are these us and other multicellular organisms. Oh, this is too small. Which one is us? Anyway, one of the red dots is us. 8 is us. We're not very good. And then the blue and the green are single-celled organisms, eubacteria and unicellular eukaryotes. So despite the fact that we're all as complex and so wonderful, we think, we're actually not really great at repairing our DNA. And furthermore, our genomes are huge relative to bacteria. So multiply this up by a much larger genome, and we suffer so many more errors in replication than do single-celled organisms. Anybody want to venture an explanation for why we're so bad and they're so good at repairing their DNA? What was that? Well, in eukaryotes, you have recombinational repair from the other template, but that would work in the other direction. It should make repair better in diploids. Sorry, in multicellular eukaryotes. Generally, I think prokaryotes have faster cell replication times. The minimum cell replication time for E. coli, I think, is like 20 or 30 minutes. So it's really quick. But that's you're getting to the proximal mechanism, which is what do they do? How much time are they spending error checking their DNA? So I'm not going to spend too much time there, but is there another guess? Yeah, I think you're talking about what happens to the mutations once they occur and whether or not they're weeded out of the population due to selection.
Yeah, and hence
Yeah. Yeah. And eukaryotes. Yeah, it is true that eukaryotes have these larger genomes, and so probably more sites are neutral. I think that's your argument. And because more sites are neutral, maybe there's just less selection against mutation pressure. So is that nice? Nice. That's a good explanation. The one Lynch proposes is that it's actually got to do with Mike's lectures, population size. The bacteria and the multicellular eukaryotes, some of which have large genomes and they have introns and whatever, but they have huge population sizes. So the effective population size is so large that weak selection can still influence evolutionary change. We haven't really talked about it, but I'm gonna, Kavita mentioned a formula for the fixation probability of a mutation. This is in haploids. And she mentioned that it kind of goes to 2s when s is much larger than 1/n, and it goes to 1/n when s is 0. And kind of in between, you can show that when s is approximately the same as 1/n, that's a good cutoff, a good rule of thumb. Selection has to be stronger than 1/n for selection to dominate this equation. And if selection is less than 1/n, it's pretty close to the neutral expectation of 1/n. And you can plot this and demonstrate that to yourself. And so Lynch's argument is that the population size of unicellular organisms is so much larger that there's a larger range of selection coefficients that are effective. And in particular, those kind of small fine-tuning of the polymerases and the other molecular machinery that repairs their DNA is more efficiently, selection acts more efficiently on that. And so this is Nature Reviews Genetics. He outlines his argument for that. But I think your explanation was actually the preceding one. And I think his evidence is pretty strong, but I wouldn't say it's 100% nailed down. So, okay, so viruses, they're not on this plot. Viruses are kind of hijacking the host repair. And so depending on the type of virus, if it's a DNA virus that incorporates like HIV into our genomes, it has the same mutation rate pretty much as the host. The RNA viruses, they're ones that have no error checking, and they have very high mutation rates. Then there are viruses like COVID, which is an RNA virus, but it has error checking capabilities. Nowhere near as good as any of these, but still reduces the error rate in those RNA viruses. Yeah, so where would they be? I can't exactly put them on this. But much higher, I would say, for the RNA viruses, much, much higher. Yeah. And here too, notice that this is per generation, so those generations are smaller, so you have to adjust for that too. Yeah. Yes, yeah, yeah. Oh, don't get me started on that. Maybe let's come back to that. error thresholds. Can we push organisms? Can we push viruses up a little bit in their mutation rate? So much that they make no viable offspring. That's called the error threshold. And there's been a drug that's been released, approved in many, many countries around the world for COVID called molnupiravir. And that's where I get on my soapbox, and I'll stop my soapbox right now because, yeah, we'll talk about it after. All right. So just to get commit these numbers roughly to memory, that in eukaryotes, those point mutation rates per generation are roughly 10 to the -8 to 10 to the -10. So they're low, 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 low. So we've evolved to be pretty good at repairing our DNA. Of course, that's per site, and then per genes, I don't know, on average about 1,000 base pairs, so it's 10 to the -5, 10 to the -7 per gene. I am going to mainly skip this because Kavita talked about the balance between mutations in one direction and in the other direction, in the absence of selection. She was looking at mutations in drift, and I'm just showing you, I think that she had this equation already drawn up, that in the next generation, you're going to get the big A allele if you had the big A allele before and it did not mutate, or if you had the little A allele and it did mutate. And here, and in general, backward and forward mutation rates don't have to be equal to one another, especially if you're talking about something like an insertion or a deletion or what have you, because or even a SNP. If you have a mutation to any of the other 3 nucleotides at a site, that's 3 times more likely than the reverse mutation to that exact back mutation. So back and forward mutation rates don't have to be equal. You can solve for the equilibrium, setting the 2 equal to one another. It's a half if the forward and backward mutation rates are equal, as Kavita showed. But if you just solve this equation by setting P to P hat, an equilibrium, I'm gonna just do that. I can't just talk to you about it and not do it. Everywhere that I see P, I'm gonna write it with a hat on top, and that represents an equilibrium. That means there's no more dependence on time. It's just at a point, it's going to stay there. So I've got P hat is equal to 1 minus mu P hat plus nu times 1 minus P hat. And so when we have a recursion equation, it's mapping from one time to the next time point, and we have different times on the left and the right. But now these have the same equilibrium concept on the left and the right, the same P. So all we have to do is gather the P's together and solve. So this is just expanding. Help me simplify that. I want to solve for P hat. Anybody willing? If you see something that's simple, do the simple thing, and then we'll move to another person. I want to solve for P hat, but I've got P hats on the left and P hats on the right. And I know you can do it because you did it for me yesterday, I think. Yep. Yep. Okay, so let's subtract subtract this from both sides. Subtract this from both sides. So minus UP will be a plus, add it to both sides. Subtract nu from both sides, add nu P hat. Ooh, this one I want to keep over there because that doesn't involve P hat. Add nu P hat to both sides. And on the right, the only thing I have left is nu. Okay, next, who wants to simplify this next? Yes. These cancel out. Always look for things that cancel out, and that leaves me with and then divide both sides. So that's isolating. So any linear equation like this, where every variable, the p's only enter in as p's, not as p squareds or cosine of p's or anything complicated, can always be solved using this method. You move everything involving p's to one side, factor, divide both sides by that factor. A couple of key points. Just so if we have an understanding how a system changes from one generation to the next, we can always ask, when would it not change? When would it come right back to where we started? And that's basically what this calculation does, is it says, if we were at time t at this equilibrium, we have to come back to the very same point. So I replaced pt plus 1 with this equilibrium, and I replaced p of t with this equilibrium. And so it's saying what would have to be true for us to return back to the same point if we started at that point. And that's how you find equilibria in general in all of these models. And that is plotted here, mu over mu plus nu. But remember what I said mutation rates were, they're teeny, like 10 to the negative 8 for site, or 10 to the negative 9. So look at the time, and this I jacked up the mutation rates, maybe representing the gene-wide mutation rate. But look at how much time it takes to get there. So yes, it will reach this mutation balance, but it takes an awfully long time. And the forces of mutation kind of pushing the system in are weak. They're weak relative to drift, and they're weak relative to selection most of the time. Okay. We want to focus on those that have an effect on fitness. So while many mutations have no effect on fitness, those that do tend to reduce fitness. They tend to be deleterious. And so here's just a kind of a cartoon image of you have two alleles in the population. Selection is favoring one type, the non-mutant type, big A, but it's continually mutating to little a, but little a is less fit, so it's being eliminated from the population. And so we want to ask, what's the balance? How many of these do we expect to see in a population between this input of mutation and this outflow due to selection? But before I do that, I want to just say, what do we know about these selection coefficients? And what do we know about these genes that are causing some of the problems that we have in the human population? How many of you were alive in the year 2000? Oh, actually, a lot of you. That was when the draft human genome was published, and there was a lot of fanfare. We're going to be able to solve all of the diseases, and we're going to be able to fix everything, and we're going to know everything. And I want to look at this timeline. This is the right shows the timeline of figuring out anything about complex diseases in humans. And you can see there's like nothing. It's changed recently. We did learn over time about the genetic basis of several kind of simple Mendelian disorders. That includes cystic fibrosis was learned in this way. I'll have a slide in the next slide. I'll talk about a few of them. But for the complex ones, for things like depression or obesity or diabetes, all of those, we really didn't learn much for a very, very long time. Anybody want to guess why it went up recently? What was that? Sequencing. It wasn't enough to have one genome to figure this out. The only insight was gained once we had so many sequences in a population that we could actually do things like associate these quantitative traits with what are the SNPs or alleles that individual carries. So it really wasn't until the genomes went through the roof and at a population-wide level that any insight into complex diseases started to happen. And it's made a huge difference. It's allowed, so it was a delay, this promise was delayed, but I would say that we have now really improved targeted medicine based on this genomic information. And that includes the kind of treatments or actions for a number of diseases, cystic fibrosis. Once the gene was known, Evacator was developed. Fabry disease, Miguel-Stat was disordered, so targeted therapeutics. In other cases, phenylketonuria or Usher syndrome, it's just avoiding certain things in the diet and you're fine. Was there a question there? Okay. So we are improving our understanding of the genetic basis of human diseases and as a consequence, getting better in targeted medicines. One of my students suffered from lymphoma. He almost died. And one of the first things they did was they sequenced his genome and figured out that he had a particular translocation. And so they could type his type of cancer and target his immunotherapy at a genomic level for an individual. He's alive and it's 5 years on. So I am just grateful that he's here. All right. So that's what we're learning more and more about mutations. We still don't know a lot, but let's get to the modeling. And as I mentioned, we are now adding a second process. We have, I flipped this around relative to, this is gamete union. I knew I did that. Diploids at the bottom, meiosis there, haploids on top, and I'm going to talk about haploid selection mainly. But we've already modeled haploid selection. This is the equation that I wrote up on the board for how the types change from one generation to the next due to selection. So after about a selection, that's what we have. But now we're going to add mutations to this. And we're going to add mutations during meiosis, during the production of gametes. This is a little bit not true. Mutations are constantly occurring over time, but models tend to simplify it and just place them somewhere. It doesn't make much difference if you move the mutations around, not much. But just in case you were wondering. And then this is the same equation for mutation that we had before, and all we have to do is kind of nest them. If in the life cycle first selection is happening and then mutation, all you do is plug in this equation after selection into that equation and you're done. But we're not done, because I like to prove things. And so I'm just going to give you an illustration of how to prove what the balance ends up at. It's kind of intuitive, so this is a bit of a long-winded thing for something that's so intuitive, but there you have it. All right, so this is what I want to do. I want to do this nesting, and then I want to see where it will, what will the equilibrium be? That is, if we start at that mutation selection balance equilibrium, we'll return to the exact same point. That's what I want to find, that point. And while I'm doing this proof, part of why I want to do it is so that you get the flavor of what it's like to build up these evolutionary models. Because it's just kind of like you do this, and then you add your process of interest. But these building blocks are basically what you use in more and more complicated models. And so we're adding right now a building block of mutation and a building block of selection and putting those together to understand how the two interact. Okay. So what I'm going to aim for is to try and find the frequency at equilibrium of the deleterious mutation. So this is the disease-causing allele. That's what I want. And I'm going to assume that the fitnesses are measured relative to the wild type, and that selection decreases fitness in the little a allele. Warning, warning, warning. Selection sometimes is defined this way as a deleterious effect. Sometimes I've already used it as a 1+ S, as a beneficial S. Selection, you always have to pay attention to the paper. How are they defining selection? Sometimes they're even defining selection in the heterozygotes or in the homozygotes. Always pay attention. S has no standardized meaning. Okay. Here's today's definition of S is the deleterious effect of the mutation. Okay, so I'm going to rewrite all my P's in terms of Q's. And again, I'm interested in what value of P and Q will return me to exactly the same value in the next time step. So I'm going to replace those t's with those p hats. So p hat, which will equal 1 minus q hat, because I'm trying to solve for q hat, will equal 1 minus mu p hat after selection plus mu q hat after selection. I can tell you that the math can be expanded. You're going to end up with a quadratic equation that you're going to have to try and simplify. But I'm going to make the math easier by saying, mutation rates are so small. Anything that's kind of proportional to the mutation rate squared, let's ignore and just drop out. So we're going to pay attention only to the leading order terms. Anything that's mu squared, drop. Mu nu, drop. And moreover, this allele frequency, we'll see it at the end, it's true. It also is proportional to the mutation rate. No mutations, it's 0, because selection acts against it at equilibrium. So this one's proportional to the mutation rate. So anytime I see something like Q times the mutation rate, that's also going to be small squared, really, really small, and let's ignore it. So this term, mutation rate times a mutant frequency, is really small, and we're going to ignore it. This one here, I want to expand out, and I also want to replace P with 1 minus Q, so we get 1 minus mu Q hat selection minus Q hat selection plus mu Q hat selection. And I can do the same thing here. This part here is a mutation rate times the mutant allele frequency. That's really small squared, and I don't want it. On this side, I have a 1, and on that side, I have a 1. I don't need those. I can drop them. Let's move everything involving Q to one side and everything involving mu to this side. So I've got mu adding Q to both sides. Q hat, that gets cancels. And then I get minus Q hat selection. Whenever you're modeling, I haven't plugged in yet my P selection. I haven't used this formula. And it's just generally a good rule not to bother until you need it, because then you keep your math clean until you need it. We need it now. To get any further, I need to have my Q selection. So we have Q hat, and then we have Q selection is, the P selection is this. If I did 1 minus P, I'd get Q, and the formula for that Q would be the little a's fitness times its frequency. Now I'm doing at equilibrium over the mean fitness. I could do 1 minus that and expand, but instead I'm just going to tell you that that's the equivalent for the little a allele of this equation for the big A allele. Just swap big A's and little a's, P's and Q's. You get that equation. Okay. But sorry, and then W little a is 1 minus S. Line, make it easier to see. Now, W bar has all of these terms in it. But I'm going to, when I multiply by W bar, I'm going to multiply W bar times mu, and I'm going to multiply W bar times Q, both of which are small. Mutation rate and something that's the mutant frequency is small. So anything that's small, mutation rate, and then the mean fitness, I can ignore. So the mean fitness is itself W big A, which is 1 times P hat plus 1 minus S times Q hat. I can draw those together. That's P hat plus Q hat is 1 minus SQ hat. And my point is that I could multiply this through and then drop things that would be like mu times Q hat, or I could just say, let's drop them in the denominator, because I'm multiplying on my small things and they'll get dropped in the next step anyway. Either which way, the mean fitness times mu is approximately mu. This one, Q times the mean fitness is approximately Q. And then we have minus 1 minus S Q hat. And if you're not comfortable with that, just go ahead and multiply by the mean fitness. But then you can scan these terms and say, oh, well, Q hat times mu is small. It's of the order of mu of the mutation rate squared. So we can ignore it. One last step, there's a mu here, there's a Q here, a minus Q there. I don't need that. And I don't need that. Those will cancel. And then we have a minus minus SQ or just SQ hat. So I've divide both sides by S and I get my answer. The mutation rate over the selection coefficient is the allele frequency that the mutation will go towards. And sorry, I haven't proved that yet. That if I start there, I stay there. And I can prove, but I won't, that it actually will approach this value. It's a stable equilibrium. To some extent, I did a lot of math, but it's intuitive. Mutations are introducing these mutant alleles, selection is weeding them out, so it makes sense that they would approach a balance. I also assumed along the way that this mutant allele frequency would have to be rare if the mutation rates are rare. And this is self-consistent because the answer is actually a small number if the mutation rates are rare. So that's good. It depends on mu only and not nu. Why is that? Yeah, exactly, exactly. Good. All right. And if you want to know more about how do you prove stability in these systems, how do you do perturbations to know where these systems are going, then I can recommend a book. And we're trying to figure out whether the book is on the library or not. Otherwise, I'll post the PDFs. But we'll figure that out. Okay. So we find the mutation selection balance, it's called. And that tells us how large this pool is going to be. So the key point is that if mutation repeatedly generates deleterious alleles, then they will persist in the population despite their negative effects. Selection will remove them, but mutation will reintroduce them. What if the mutation is lethal? What's this answer? How many deleterious mutations will we see in the population of babies? S is 1. After selection, you're right. But before selection has acted, then S is, what's the number of, for fitness of the little a allele is, let me go back. We're saying little a has a fitness of 0. It's a lethal mutation. What does that make S? 1. What does that make that frequency? Mu. What does that mean in terms of, you're right, they're going to be dead. So at the end of the generation, there won't be any. But at the beginning of the generation, we expect. The new mutations that have just risen in that generation that are proportional to mu. So every generation, you're going to get this kind of new input of mutations for sure. Good. Mean fitness. I'm going to skip this, but it'll be in the slides if you want to look at the mean fitness over time. But I've already talked about how if you started at all big A's, you're going to have a decrease in mean fitness. So we've already talked about this. The other thing that's a quick calculation is if I calculated the mean fitness, and we have it on the board, I already did it over here. The mean fitness is 1 minus SQ hat. And I plug in Q hat is mu over S. S's disappear. The mean fitness is just 1 minus mu. It doesn't depend on the selection coefficient. This is sometimes called the mutation load. It's the amount by which the fitness of a population is reduced by mutations that are recurrent. Why does it not depend on S? And maybe the math will help you think this through, or maybe you've got an intuition for it. What was that? Yes, so this is a mean fitness, 1 minus SQ hat. It's the amount by which the fitness is reduced by selection times the frequency of the mutant. But if I plug in Q hat equals mu over H over S at the mutation selection balance, then I get 1 minus S mu over S the S's cancel, and the mean fitness at equilibrium is 1 minus mu. It does not depend on S. Why does it not depend on S?
That selection is how rapidly the mutation, but still the mutation rate, regardless of S.
Yeah.
Yeah, the thing would be constant.
Yes. So if S is really high, it's very lethal mutation. It's weeded out very, very quickly. And it's at a low frequency because it doesn't persist for a long time. It's weeded out quickly. So the frequency is low for if S is really high. But then it has a fitness effect of S. So you multiply it by its fitness effect, or this cost. But this fitness cost exactly balances the fact that it's rarer. So really bad things, they don't persist for long, as you said. They reach a really low mutation selection balance. But every time you carry them, you have a really bad severe effect. But the severe effect exactly cancels out this shorter residency time of the deleterious mutation. But sometimes you just have to do the math because it's not always intuitive. And that's at one locus. So key point, mutations reduce fitness on average by an amount that depends only on the mutation rate, no matter how strong selection is. And then this is the reasoning we've just talked about. And I'm gonna I wanted to focus on the haploid model. I like the haploid model because you can really do more of the math and I can show you what's going on. But the diploid model is equivalent. We're just gonna add mutations in following selection that we already did on the board this morning. So in particular, it's standard for deleterious mutations to be described by these selection coefficients and dominance coefficients. So as I mentioned, you always have to pay attention to what the context that selection is defined in, but this is a pretty standard one for deleterious mutations in diploids, where if it's homozygote, it reduces fitness by S, but if it's heterozygote, it reduces fitness by some intermediate amount, and that H is the dominance coefficient. Dominance coefficients tend to be between 0 and 0.5. Mike has a review paper on this in Tree, I think. So most deleterious mutations are partially recessive to fully recessive. H is less than 0.5. And that's thought to be because when many of these mutations are breaking something, breaking an enzyme, making it non-functional. But as long as you have a functional copy, you're okay. So anyway, it's not always the case, but oftentimes the heterozygote is closer in fitness to the homozygote than it is to the mutant homozygote. We already have this equation from the board underneath here saying how selection would change things, and you can already start to see, what do? I put that building block to the other building block for mutations that we already know when mutations are entering, and do the same kind of nesting. I will save you from doing this on the board, but feel free to nab me and I will do it. And what you find is actually very, very similar. Let's start here. That there's a similar mutation selection balance that's reached, but it's mu over HS, not mu over S that's reached. And the reason for that is even though the fitness of the diseased homozygote might be really, really low, it's rare. Most of the time that the mutation is present, it's only present in the heterozygote. And so it's the fitness of the heterozygote, the cost of the mutation in the heterozygote that really matters. But still, you reach an equilibrium that's mutation selection balance. But clearly this can't be right for H is 0 or H really near 0. If it's fully recessive, this can't work. So you have to do the math again. And if it's fully recessive and H is 0, then you do the math again and you find a different equilibrium. But the method is the same for finding that equilibrium. Question? So one of the key points from that, so remember, mu is really, really small. So if it's got some expression in the heterozygote, mu over HS is proportional to this really small thing, mu. The square root of mu is a larger number. right? Because you take 10 to the negative 8 and you square root it, you've got 10 to the negative 4, much larger. And so this leads to the prediction that recessive mutations should be found at a much higher frequency within a population because they will persist for a lot longer. They won't be weeded out as heterozygotes, they'll persist, and only be weeded out once they are found in homozygous form. And so this is just a histogram showing the difference between basically the frequency with which we find dominant mutations versus recessive mutations in different categories. So, for example, singletons are much more likely to be recessive, 2 to 10 copies much more likely to be recessive, and higher frequencies much more likely to be recessive. By contrast, dominant diseases are much more likely to be absent from a population because they are subject to selection when they first appear. So that's one prediction from these models. If you do the same, so this is the mean fitness in a diploid population at equilibrium, p squared w big A, 2pq w big A little a, q squared w little a little a. And if you plug in all the numbers, and again, ignore mu squared terms, you get 1 minus 2 mu. Again, the selection doesn't matter. The stronger selection is against the deleterious mutation, the worse it is for you. You don't want that disease, but the disease will be rare in the population, and those exactly cancel out. And you get to a load of 1 minus 2 mu. That's at one locus, and I just want to briefly point out that it's thought, of course, that that's happening across the whole genome. And so if you take the load due to deleterious mutations and you multiply it at one locus times the next locus times the next locus times the next locus raised to the power L. And I'm just going to tell you, if you're not familiar with this, but 1 minus a small number is really nearly e to the minus a small number. So this is a close approximation. So this is helps us get a handle on how much load do we expect in a genome, a eukaryotic genome. If there are L sites subject to selection, it's diploid here, and the mutation rate per site is mu. And this quantity 2 mu L is often collapsed into a quantity called the genome-wide deleterious mutation rate, which is the subject of many experiments aimed at estimating that mutation rate, as shown here. And so the citations are here for the different mutation rates, but the genome-wide deleterious mutation rates is thought to be pretty high, actually, in humans and other species. Mike, what's the latest deleterious mutation rate for the human genome? What was that? 2.1. Okay, even worse. So if you plug that in, that means the mean fitness, we should be really like dead, walking around with a lot of deleterious mutations. But we're not all dead. We're actually mainly fine. So this concept of the mutation load has actually led to a lot of kind of speculations about where this load is happening, or maybe if it's happening at all. So for example, maybe it's happening at the gamete stage or the zygote stage. Many zygotes don't make it to term, and so that could be a lot of this mutation load experience at that stage. I'd be dead, and my son would probably be dead too, if in the absence of medical care, because we have problems during birthing. And so medical care is actually eliminating a lot of selection, thank goodness. So maybe some of this load is just kind of temporarily reprieved due to medical practice. But this type of issue, how large is the genome? How much deleterious mutations is it suffering? Is a big issue in conservation biology, because if species have recurrent deleterious mutations, then you think you might have a large population, but actually they may not be as fit. Mike, do you want to comment any more on that? Because this is something you've worked a lot on.
Well, just one thought, which is another component of fitness that I don't mention is country.
Yes.
And so if we just don't have this, in order to imagine an unloaded individual human, you only only have to imagine somebody that's having about 50 offspring. And that sounds like a lot, but the world record for the number of offspring for a woman is 69. So it's clearly possible for a human to have the amount of offspring that supplies an unloaded genotype. And so it's important to think that what we're used to in terms of thinking about what humans are like or what software is like, we're thinking about the loaded population. We're not thinking about what it would be like if we didn't have a military. And if you do, then it's all consistent.
Yeah.
Necessarily puzzled.
Yeah. And so mutations are really a good news, bad news story. They are the source of many deleterious diseases that we and other species suffer, but on the other hand, evolution would grind to a halt. We couldn't get evolutionary rescue without mutations. We couldn't adapt to new environments, to climate change without mutations, but they also come with an expectation that they will reduce average fitness in most populations. Yeah. What was that?
RNA viruses, for example, will have
Yeah.
a huge mutation.
Right. So the mutation rate is like 0.5 per genome per generation. So it's not that large. Yeah. And that was just for COVID. It mutates once per genome every second generation, roughly. Yeah. Okay, so I have some worked examples, but I think you can do them about, so the balance between mutations and selection in human populations. Initially, this was the first way that we got at mutation rates at all in humans and in other species, was actually using this math to back calculate what the mutation rate must be, given what we were observing as the fitness effects of mutation. So there's a couple of worked examples, one for a recessive condition and one for a dominant condition towards the end of these slides, but I'll post the slides and you can work through them yourselves. So I could get back on my soapbox or stop there or take questions. What would you guys like? Yes, so it's a beneficial mutation, but here's one way I might be modeling it. Big A is my wild type, little a is my mutant, but now rather than bad, it's good. But I might have, I think, before we said, oh no, big A is your new mutation, little a is your resident mutation. Now I'm gonna flip it like this. As an example, it's arbitrary. And you have to specify who's your reference and who is your new type. Yeah, so be careful with S. And as I mentioned, for the diploids, it's even nastier because maybe Kavita and Mike can add more to this, but I'm just going to add to all sorts of ways, conventions that I've seen to us. So this is an additive assumption. That's actually making an assumption. But I've seen 1 minus S and 1 and 1 minus T. I've seen 1, what was that? 1 minus HS and 1 minus S. And so, and the reason, actually the math works out easier in different cases depending. So there's a reason to choose your notation and change it, but just be careful. Any other questions? So tomorrow's lecture, I have a little bit of wiggle room in it. So if there's something that you're like, I've never understood this idea or concept or proof or anything, then send me a direct message in Slack, because I can modify my lecture for tomorrow easily. And can I get on my soapbox for a minute? 1 minute. So Kavita asked this question, where are these viruses? Where is COVID? And COVID is kind of in the middle here. And the idea of the error threshold is, well, let's push them, their mutation rate so high that they can't replicate any virus. Molnupiravir, produced by Merck, is a nucleoside analog. What that means is rather than ACGU, it substitutes in. And I can't remember exactly which mutation, but it tends to cause Cs to mutate to Gs or Us. It's a little bit of a wobble there, but it creates mutations. And so the RNA should be peppered with mutations. It actually doesn't know what it's incorporating into. This nucleoside can also be incorporated into our cells. So that it can be a mutagen as well for us, not just for the virus. So the idea is if we could just jack up the mutation rates so much that they have no viable virus offspring, we can cure ourselves. But when Merck produced their data, what did I want to see? I wanted to see a virus riddled with mutations. It had like 2 more mutations in the genome. It wasn't riddled with mutations. And if you think about a drug permeating a body, it's not going to get everywhere at high enough dose to mutagenize every single cell. So reading that data, I was like, this is really in the bad zone, where it's increasing the mutation rate, but not obliterating the virus, it's just increasing it. So we're just going to be adding mutations in, and that's a risk. It also wasn't helping people that much, and the original data was unclear. This now, is it a year and a half after, 2 years after it's been approved, it's shown that it reduces the course of your disease a little bit, but it actually doesn't prevent hospitalization or death. And so for that reason, the fact that and it has been shown that the mutations that are generated by molnupiravir are now, there's signals of that in the genome of SARS-CoV-2, only in countries that have approved molnupiravir. It hasn't led, as far as I know, to any one of these new variants of concern, but we basically added fuel to this fire by increasing the mutation rate, and particularly rare mutations like Deepa studies for this virus. So that's my soapbox. Oh, it's even worse. What's even worse? Molnupiravir is, they went, what's it called when a drug is not, a coffee time. A drug, it went cheap. So now molnupiravir is basically the only cheap option for therapeutics, despite the fact that it has this danger. What is it? Paxlovid? Yeah, is another drug treatment option, but it's like $1,000 a course. Anyway, yes.
Yeah, I was thinking because if I remember correctly, there was this problem with the error threshold because completely large populations versus the finite ones in which is smallest ratchet, right?
Yeah.
And basically you reach some kind of mutation selection balance. So you cannot really push it through error thresholds. I was just wondering that if you use these therapies, when you're thinking of smallest ratchet type of thing, that means you will have some sort of balance reached, or is it?
No, it's really this deterministic model that you're basically, so a virus has to make one new disease per disease. And if you can just reduce the number of viruses and drop the amount of virus so much that it can't replace itself, that's what the goal is. So it's a deterministic process. But it treats us as one big sack of viruses without accounting for spatial variation and so on. Anyway, Mike is going to coffee and he's going to drag us all out so I'm going to stop. Thank you, everybody.